My name is Tobiasz Druciarek. I am an acarologist. I study mostly mites, especially those that transmit plant viruses. I try to focus mostly on aerophyte mites because they are understudied and they transmit plant viruses. I study rosette virus since 2011. We are facing an epidemic of rosette in the United States right now, and situation is getting worse each year. And it's really big problem for the rose industry. So rosette disease is a deadly disease of roses. And it infects only rose plants, and it spreads through a microscopic, tiny little mite that is blown by wind from one rose to another. And those mites carry the virus inside of them, and when they feed on the new, healthy rose, they transmit this virus to a new plant that eventually kill that rose within 2 to 5 years from the infection. For 70 years, we didn't even know what's causing rosette disease. This changed in 2011 when the causal agent, rosette virus, was identified in our lab in Arkansas by Professor Yannis Tsanetakis and his student. And Phyllocophtes fructifilis is a confirmed vector of rosette virus. I studied the population structure of aerophyte mites on roses to really see what other species might be involved in the spread of this disease. And then I also studied if it's possible for this virus to replicate, what's the mode of transmission, if it's possible to replicate inside the vector. And it turns out that yes, this virus is replicating inside the mites as well, so not only in the plant. I have also found a second vector of that virus. We described it and named it Phyllocophtes arcani. So symptoms may vary depending on the rose species or even cultivar, but typically they include long-lasting reddening of those shoots, distortion and proliferations, and very characteristic excessive thorniness. So there's a lot of small tiny thorns, and also characteristic dense structures are formed called witch's broom. Hopefully in the future, as we more know and understand interaction with the vector and with the plant, we will be able to devise control strategies that could be applied in the field. But right now, prevention is the only option. We don't really know what miticides or acaricides are effective against the vectors of rosette because nobody studied that. And the problem with those mites is also that they are hiding on the plants. Because of their microscopic size and lifestyle, they are a refuge-seeking type of mites. They developed and occupied places on the plants that are hard to reach with conventional pesticides. Inside the flower buds, under the bud scales, at the base of petals, in crevices, in places that, you know, even when we spray, it's really hard to control populations of those mites. So right now, prevention is the only thing that is being recommended for rosette. So scouting for infected plants, symptomatic plants, and quick removal of those plants. But prevention is not really a solution. I hope that we will be able to develop practical control solutions to control vectoring mites in the field. That I think is the most important. I hope we will finally, understand better how the virus interacts with the vector, what are the molecular mechanisms of that interaction, so that knowledge can be used to devise really effective control strategies for this outbreak that we are facing.